Davis, I'll turn it over to you.
Thank you so much, and I am so excited to be here. We go way back, right when I was ripe in my postdoc training, and as I'm sure Dr. Hughes-Halbert can attest to, sometimes, you know, when we were early in this sort of realm of trying to identify basic science mechanisms behind disparities, sometimes, you know, we were met with a little bit of resistance, but, you know, over the years, I think we have definitely sort of collected a large body of evidence, still collecting more. And in our attempts, you know, some of the shortcomings of, even though we've had, you know, numerous advances in technology, if you don't have sort of the inclusion, right, of a multi-ethnic population in these research endeavors, you won't be able to identify some of the things that our groups have been able to discover. So thank you everyone for coming. Today I'm going to talk a little bit about sort of the spectrum. I'm going to give you a brief introduction about how the disparities that I study, how we believe they even emerged. They are dynamic, meaning that, you know, the disparities that we talk about today are different from the disparities that we saw or that we would talk about, you know, 10 years ago or 5 years ago. But what I believe is true, I have no financial disclosures, by the way, unfortunately, maybe one day. So this is the outline of what I'm going to talk to you about. But what I think is true is that we know that as our environments change, so do our risk of cancer in general. And what I'm going to start talking to you about is just how, you know, breast cancer disparities have emerged, what I mean by disparities in the context of the research that I do. And then I'm going to give you some background about distinct tumor biology. And then I'm going to share some emerging data, some of which we published, some of which we're trying to get out there. So, you know, given the audience here, I'm sure you already are aware that breast cancer mortality is more than 40% higher for African American women in the US than Caucasians. And that wasn't always the case. So prior to the mid 80s, so the graphic, can you guys see my arrow, by the way?
Yes, yes.
Okay, so let me orient you to this graph. So the first two lines here actually represent the incidence of breast cancer. The top line represents white women, the red line represents Black women. And this is data from SEER. And it actually shows that white women traditionally, since the early 70s, had a higher incidence of breast cancer compared to Black women. And we start to see this line converge in more recent years. And so this convergence basically tells us that the risk for breast cancer, though it seemed like it was higher in white Americans, actually isn't necessarily higher in that group. So some of this is because we do a better job of screening, a better job of just surveillance across the United States population in the tumor registries. But basically, when we look at then the incidence of, or the mortality rate, excuse me, so now these two bottom lines actually represent mortality. And so the red line represents whites, the purple line represents Blacks. And so what we see is that prior to the early 80s, it really didn't make a difference if you were Black or white. You had pretty much the same chance of surviving breast cancer. But what started to happen as we see this gap start to emerge, this is the mortality disparities. And what happened was that we see this red line start to decline at a higher rate, which means that the mortality in whites was dropping at a much faster rate, which is good, right? We want to see lower mortality. But in Blacks, we don't necessarily see that same rate of decline of mortality. So if we actually look at what was happening concurrently to when this line started to diverge, we'll see that this is also the timing of the first endocrine targeted therapies and the timing at which targeted therapy was standard of care. And so what do I mean by targeted therapy? I mean that instead of just having a general systemic treatment, sort of like chemotherapy, for the tumors, we actually had molecular markers and we were create drugs that could target those molecular pathways because we understood that there were hormones driving the sort of growth of the tumor. And so we were able to, the proverbial we, of course, were able to identify drug targets. And so these drug targets were absolutely phenomenal. I mean, now we can even, depending on, you know, how early you diagnose breast cancer, it's not uncommon that women would be cured of their breast cancer. However, this therapy or access to therapy was not as beneficial to Blacks as it was to whites. And so I cannot really talk about disparities in the outcome without, you know, actually paying attention to the disparity in the causes, right? And so when we look at cultural differences or social determinants, if you will, of healthcare in general, we know that environment plays a very key role. So when we look at differences in whites versus Blacks in the US, we know that socioeconomic status often determines your external or built environment. That environment then precludes you to, you know, your housing situation, pollution in your area, your access to nutrition, your access to healthcare. We also have cultural traditions of the way we eat, our physical activity. And all of this can potentially translate into some biological pathway, right? So where you live, where your exposure to pollution also means you're exposed to mutagens, and mutagens are what damage DNA and what typically drive or cause tumor formation. And so I'm not going to spend a lot of time on this because my group doesn't necessarily focus our lens, if you will, on systemic racism or, you know, the cultural environment that precludes certain groups to getting cancer, but more we are focused on the outcome. So once you have cancer, how can we better treat this population with targeted therapies? But in thinking about sort of a genetic predisposition to certain types of breast cancer, and I'm going to get into the different types in a moment, we know, for instance, that BRCA gives you a higher risk, but we also understand that cancer is a complex disease. And so when you think about a complex disease, meaning that there are multiple factors, so your environment, which we just touched upon, we also know that there are multiple or polygenes that are it's a polygenic disorder, meaning that more than one gene has a significant influence. And so you actually have to have a combination of, you know, variants, if you will, to have the effect of actually having a tumor. And so we know that environment does have an interaction, because if it didn't, then, you know, it really wouldn't matter where you lived. We also know that, you know, even though socioeconomic status does preclude certain race groups to overexposures, you know, due to structural environment, we also know that other races also live in those environments, but we tend to see more of certain types of tumors, particularly in people of African descent, regardless of their environment. And so as we talk about BRCA, you know, one way to identify a genetic risk is to follow the transmission of that disease, if you will. Now we're talking about breast cancer. Follow that transmission through family groups. So if it really is a genetic predisposition, then we should be able to see it transmit genetically through offspring of individuals who had the disorder. So this is a pedigree. It's actually one of the pedigrees that Marie Claire King, pictured here, utilized when she and her group identified the location of the BRCA gene in the human genome. And so where you see the shaded circles also represents people who have, in this case, breast cancer. So you can tell right off that these are large families. Some of these would go back 4 or 5 generations. Okay, and so really you need that depth of genetic inheritance to study in order to really follow a molecular marker. And the same is true today, right? So if we were going to identify a new gene that was particularly virulent, for instance, in African Americans, then we would have to have these pedigrees. And unfortunately, this level of information is not available in the African American community, for one. Typically, we don't have pedigree information that goes back more than 3 generations. And even if we did, we don't have these annotated pedigrees with very detailed and accurate information about health history. And so we then have to on large population studies in order to identify a common thread, if you will, of shared genetic ancestry, to identify what we think is driving a predisposition. And this isn't a new concept of ancestral susceptibility, and I'll get into that a little bit more. But then, like I said, we focus on survival, right? And so just like there are multiple components to getting cancer, there are also multiple components in surviving cancer. So when we think about racial differences in survival, we have to look at all of those factors at once. And so a few years back actually did this multivariate analysis, trying to identify the components of these multiple variables that gave the most, if you will, contribution towards surviving breast cancer, or the difference in surviving breast cancer. And so when we look at all of these mediating factors combined, and so we're showing here the percent sort of contribution of these factors in their model. You can see that the range varies, so that means that for any given, set of people, there are varying effects of these. But what was interesting, for instance, is that we hear a lot about BMI having an effect. So, obesity has almost a 20% impact on survival, or differences in survival. But, things that we talk about within a socioeconomic strata, like insurance type, didn't necessarily have as significant impact as obesity in this study. But we also know that stage of diagnosis, so this is almost a third, contribution of disparities, is because African Americans and people in marginalized healthcare systems tend to be diagnosed at later stages. And this is just because of limitation of screening, right? So the longer the tumor grows, the later the stage you will be when you're finally diagnosed. And so, that makes it harder to treat. The longer it's there, the larger the tumor, and the greater the chance that it would spread. But what was very interesting was that also about a quarter, 24%, if you will, of the contribution of survival disparities was because of tumor characteristics. And so that really speaks to the biology of the tumor, that there were actually differences in the biology of the tumor. They didn't have the same factors in terms of what made the tumor grow. And so when we look more closely at what we mean by tumor characteristics, one of the first things that are diagnosed when a woman has breast cancer is what type of breast cancer are we dealing with. And so HR in this diagram simply means hormone receptor. And the hormone receptors we're talking about are steroid receptors. So this includes estrogen receptor and progesterone receptor. And then HER2 is human epidermal growth factor receptor as well. And so we sort of couple progesterone and ER into a hormone receptor status. And then we have targeted therapies against that status. HER2 is a separate diagnostic. So HER2 positive cancers also have targeted therapies. So we, again, if we diagnose these early enough, we can treat these pretty effectively. But in terms of this particular subtype, it's called triple negative. So that means that the tumor doesn't express either one of the hormone receptors, estrogen or progesterone, nor is there a HER2 amplification or upregulation, overexpression. And this is about 13% of all breast cancer cases. But particularly in non-Hispanic Black population, these women have the highest rate of this particular subtype. And unfortunately, there is no targeted therapy. Now, outside of just looking at these 3 hormone receptors, we have now been able to implement genomics panels where we can look at all of the genes and their expression, not just these 3. And by looking at all of the genes, we were able to identify in a paper early on a set called the PAM50. And so this is about 50 genes that allow us to identify at least 5 different types of breast cancer. And whether or not you have triple negative, so if we're looking just at the molecular phenotypes of luminal, HER2 positive, triple negative, we can also sort of identify a prognosis. So triple negative has the poorest prognosis. Luminal A, being this ER positive subtype, has the best prognosis. And again, a lot of this has to do with the fact we have targeted therapies, but some of it also has to do with the fact that, you know, luminal A tumors just grow a little slower than triple negative. And so what's also a note I want to make here is that triple negatives actually happen at an earlier age. And so as a geneticist looking at diseases, we also know that any kind of disease that has a very high component of a genetic inheritability, it would have to, or usually it happens at an earlier age. So if there's a genetic component driving cancer, then you would see it at an earlier onset. And so we know that about BRCA carriers as well. So if you have a BRCA mutation, most likely you're going to have premenopausal breast cancer, meaning earlier even than what is the recommended age for screening. Okay, so that's problematic. So not only are we not getting access as African Americans, not only do we sometimes not have access to the ability of having screening, but we also have the recommendation to start screening at a later time point than when some of these more aggressive tumors would be diagnosed. And so now I'm going to sort of shift gears. And what I think about, you know, as African Americans is our shared genetic history. And so one of the premise of my research program is whether or not our ancestral African genes play a role in cancer biology. So sort of transcending the social construct of race, socioeconomic status. I'm now focusing my lens, I'm honing in on molecular markers, whereby if we were to tomorrow have universal health care and targeted therapies, is there a genetic component that we could utilize to identify within the African American population, any population, quite frankly, that is a driver for certain tumor biology? And so now, instead of using these race groups, and so up to this point, I've really only talked about African Americans, Black Americans, white Americans. Up to this point, I've only used the social construct. But now in all of our studies, instead of just using self-reported race, and let me not discount that, because self-reported race is very informative, because it speaks to more than just, it speaks to that interaction, if you will, of cultural, ethnic factors as well. But now we're actually able, because of a project called the 1,000 Genomes Project, we're able to identify the components or the proportion, if you will, of genomes that were ancestral across the entire globe. And so this is geographic ancestry. And so based on these markers that were identified in the 1,000 genomes, there are at least 50 different human populations that we can attribute someone's genetic ancestry to. And so we quantify this now, and we measure it in all of our cohort, and we use that information to try to identify specific shared genetic ancestry. And so with the ability of quantifying ancestry, we're also able to take a more global perspective of breast cancer incidence and mortality. And just quickly, I want to point out that if we look at Africa, based on World Health Organization data, actually has the lowest incidence of breast cancer. That number is rising more recently. But overall, Africa also suffers the highest mortality rate. And so that sort of, you know, mirrors the trend that I just told you about in America, right? That white women traditionally had a higher incidence. So Black women had lower incidence of breast cancer, but a higher mortality burden. And so in steps the Center for the Study of Breast Cancer Subtypes, which is, I serve as the scientific director for this program. And one of our sort of thrusts or programmatic thrusts, if you will, is to fill in the gap. So we don't have enough information about what is different among these populations. And so We basically make sure that all of our studies are ethnically inclusive, that we have a strategic recruitment of these populations, and we are honed in on biological diversity that we would hope to translate into either better diagnostics or therapies towards ameliorating disparities. So I partnered with Dr. Lisa Newman. She's currently the chair of breast surgery at Weill Cornell Medicine. Dr. Newman founded the ICS-BCS program over 17 years ago. And she has a program where she has traveled to Ghana, usually 3 or 4 times a year. Of course, in the current climate, we probably won't be traveling that much, at least not anytime soon. But she performed surgery in Ghana, also training of clinical partners. And now that I'm a part of the program, we also help train some of our science partners as well. And what she identified was that and she also travels to Ethiopia for that same mission. And so what she noticed was that we compared patients from West Africa to patients from East Africa. She also found that patients from West Africa had a higher burden of triple negative breast cancer. And globally, I'm sorry, I didn't change this, but it's actually in press right now. We looked globally at the frequency of triple negative breast cancer, and we saw that in any population where either there's a large component of African ancestry, or if we could subdivide the population by that sort of self-report, right? So Black African Americans, Black Europeans, Black South Africans, wherever we could identify the African population or the African descendant population in a nation, we saw that that component of the population had a higher frequency of triple negative breast cancer. And so now globally, we have sort of this clue that there's something shared amongst people of African descent that gives us a predisposition to triple negative breast cancer. And so what is this shared genetic ancestry and what's the source of it? So when we think about human populations, typically we think about world migrations, and these are usually voluntary migratory patterns. And they give rise to sort of this hybrid model, which is a founder population travels and combines with some other founder population. And then we have sort of distribution of those alleles throughout this new population, right? So that's what most of these arrows represent. But in the case of the African diaspora, it was a very different story. So what this GIF demonstrates is that during the transatlantic slave trade, there wasn't just one population and one instance of a movement. This happened over the course of hundreds of years. Millions of Africans were displaced into the Americas, even from East Africa into South Asia. And this is a very different model of gene flow, if you will. And so this is called the continuous gene flow model. Dr. Rick Kittles published on this very. What's key to note here is that this African population, which by the way, is not just the West Coast of Africa, but also internal regions as well, fed into multiple generations. And so therefore we should be able to see a genetic imprint of this shared ancestry, and we do. So if we look at African Americans in the US, we can actually identify, and this is work from Sarah Tishkoff years ago, where she actually broke down the African continent based on languages for geographic reasons. And we see that African Americans actually have a combined, if you will, African ancestry. The component of their African ancestry originates from multiple regions around the continent. And so these circles across the globe represent sites where our program is currently operating collection and recruitment into our cohort. And so our overall goal, okay, is to touch and obtain information across the entire diaspora that represents this continuous gene flow. And in doing so, we would have the power to identify that shared risk that transcends the diaspora, that then gives rise to this imprint, if you will, of triple negative breast cancer risk across the globe. So one of the alleles that we study is the Duffy null allele. It arose in sub-Saharan West Africa. It's an ancestral informative marker. And it was fixed in this population because it gives immunity to certain types of malaria that are endemic in that region. If we look across the globe, we see that fingerprint, if you will, or imprint of the allele frequency that basically tracks with the transatlantic slave trade in the previous slide. And based on these markers, we also were able to identify that hereditary susceptibility for triple negative breast cancer is associated with West African ancestry. But more importantly, it's also associated specifically with the Duffy null allele, which is the top line in our graph here. And this is triple negative specific breast cancer risk, okay? And so when we think about then triple negative breast cancer, we know that not only African Americans get triple negative breast cancer. And what I'm not going to show you today are some of the survival data that we've seen and that's been published. And some of it is a little contradictory right now, but you know, whether or not white women die more frequently of triple negative breast cancer is without question. So anyone who has triple negative breast cancer will have a poorer prognosis. But in the case of African Americans, we actually see differences again in the tumor biology. So we recently published, and this was in collaboration with Dr. Clayton Yates and Dr. Main. Dr. Yates is at Tuskegee University. Dr. Main is at the University of Alabama, A&M. So we recently published a paper showing, again, where we quantified genetic ancestry across self-reported African Americans and European Americans. And first of all, we found what we expected, which was that European Americans have largely a homogeneous European ancestry. African Americans have more admixture and a range of admixture. And so once we quantified this, we were able to then correlate expression in triple negative breast cancer tumors to identify gene signatures that were unique to these self-reported race groups because it correlated with their ancestry. And so I'm sorry, I'm getting a little feedback. So I just want to remind everyone to mute yourselves. What was interesting about this was we were even able to separate out European ancestry into two subgroups, and that represents the individuals who had sort of even the slightest amount of mixed ancestry. We were able to quantify a difference in that population. And if you look at those individuals here, you can actually see that their gene signature kind of matches more with the African Americans than it does with the other European Americans. And if we look at what these genes are, and let me orient you, so the blue color means that the genes, which are different rows, have lower expression, and the green represents higher expression. And then these bars just translate what we have here, which is their supergroup ancestry makeup representing European, East Asian, American, Native American, South Asian, and African. And so you can see, based on this heat map, sort of, you know, most African Americans are admixed with multiple ancestry groups, and European Americans are largely homogeneous. But moving on, what are these genes? So when we looked at pathway analysis, we were able to identify some canonical pathways that we already know are associated with breast cancer. But what was interesting was that we didn't see sort of the polarity of that interaction for some of these. So for instance, P53 is one of the most mutated genes, and usually these are deleterious mutations, but it's one of the most mutated genes in all cancer types. And deleterious means usually it's a loss of function mutation. But in our pathway analysis, we saw that this gene was activated for some reason in African American triple negative breast cancer compared to white American triple negative breast cancer. And so we actually have a side project, which I won't talk about today, but we have a side project with Dr. Jill Barganetti, and we look at polymorphic versions of P53. And some of these are activating polymorphisms. And maybe 10 years ago, there was like this spike in publications around this polymorphic P53 being more prevalent in African Americans. And so this is all very interesting, the fact that we're converging on sort of bits of information, you know, about how ancestry, shared ancestry, gives rise to a different gene signature, these genes are potentially impacting canonical pathways in breast cancer that we already know are involved in cancer, but not in the same way that we would traditionally think about. And some other things to note here are the immune complex, other receptors, and of course chemokines, IL-6 being one that's activated. And IL-6 is a modulator of tumor microenvironment. AKT is within the pathway, for instance, insulin resistance. And so we see these themes start to arise repeatedly. Immune response has been one of the pathways when groups look at race differences in cancer. Tumor microenvironment and immune response has repeatedly come up as one of the top hits in pathway analyses. But because of our unique cohort, we can also look then at differences between East versus West Africans. And so this is preliminary evidence. Rachel, who is here today, I'm not going to put you on the spot, I promise. She's currently working on the new version of this, but our preliminary evidence actually showed that when we measured, for instance, the ancestry component of our West Africans from Ghana compared to our East Africans from Ethiopia, Ethiopians had far more admixture of European descent. And when we honed in on not just these supergroups as I've described in our previous publication, but we can even look at sort of subgroups of the continent or of the geographic regions. Ethiopians had a higher component of Italian ancestry. And so this is interesting, and a side note, in their history because they share a sort of fuzzy border with Eritrea. And Eritrea was a Italian colony for quite some time. And so this speaks to sort of their social history as well. But when we look at the differences in East versus West Africans, we also are able to find genetic signatures that are associated with ancestry. And again, when we look at the components of what these genes are, we start to see some of those same genes pop up. So NF-kappa B was in the previous figure, AKT was in the previous figure, and I'm not showing P53 in this particular diagram, but as you can imagine, we have multiple pathways that come out. And so we see the same thing where West Africans, contemporary West Africans, compared to East Africans, have sort of this activity in their tumor biology, and again, this is triple negative breast cancer, have a unique biology in their triple negative breast cancer that mirrors or parallels what we see in African Americans. I'm not going to talk a lot about this, but one of the side notes in our publication was that after treatment, we also see some unique signatures in the residual signature in the residual tumors, in African Americans, where we think there's a particular kinase that might be driving treatment resistance, for instance, in African Americans. So now I'm going to sort of switch a little bit. So I'm not going to talk about sort of large genomic studies. Now I'm going to talk about my favorite project, which is the DARC immune tumor phenotype. So I'm sure you saw the byline, and my title was The Dark Side of Breast Cancer. So DARC is the gene that the Duffy null allele resides in. And so let me tell you a little bit more about the gene. So normally it's expressed on red blood cells, and the Duffy null mutation actually removes expression of DARC on red blood cells. It was recently renamed to ACKR1, so you'll see it used interchangeably in my talk, but I usually just call it DARC. And so normally this expression on red blood cells acts as a buffer to remove chemokines from circulation. But this function is absent in people who are Duffy null. And again, it's an African ancestry specific mutation. So that means that the individuals who have the Duffy null blood group are of African descent. And so when we put that in the context of breast cancer, the question becomes, the endothelial expression of DARC still remains. And so, and I'll show you in the next slide what that function entails, but it can set up a different context, if you will, of tumor microenvironment based on genetic ancestry, right? And so when we think about the endothelial expression versus the erythrocyte or red blood cell expression, DARC on endothelial cells, which is basically the lining of blood vessels and capillaries particularly, the function is to transcytose chemokines, and these are inflammatory chemokines that recruit white blood cells normally to a place or tissue of infection. And so then the counter function then is that it would remove these chemokines from circulation. So in this way, it's setting up a gradient, if you will, that sort of hones the white blood cells specifically to this area of where infection is, and removes it from, and removes these signaling chemokines from circulation so that you don't have chronic inflammation systemically. But in the case of African Americans, we actually do see chronic systemic inflammation. And in a tumor context, tumors also secrete, if you will, these chemokines. And so what our group actually discovered was that DARC expression inside of tumors can facilitate this gradient and a higher level of inflammatory chemokines. So the question, so this begs the question, if you don't have expression, if you will, of the erythrocytic or red blood cell expression of DARC, how does that impact you systemically, and how does that impact the tumor environment? So again, we saw that we see DARC expressed in the tumor environment, and wherever DARC is expressed in the tumor environment, excuse me, we also see a higher influx of immune cells in that area. And this is Rachel, and Brittany is one of my former graduate students. She graduated last year. And I started this project in my former academic life at the University of Georgia, and these were my initial partners in that project. But last year we published that when we look at the DARC expression in tumors, looking at TCGA RNA data, so this is gene expression, the RNA level, we saw that, you know, we see a spectrum of whether or not DARC is going to be present in tumors. And when it is, we see higher levels of pro-inflammatory chemokines. So now the colors represent red as a high level of expression, blue is a low level of expression. And so tumors that have high levels of DARC also have high levels of these pro-inflammatory chemokines. And if we look across race groups, we see that primarily most of African Americans have a low expressing tumor, low DARC expression. But we also see this dynamic sort of compartmentalization of DARC across the whole spectrum of different subtypes of breast cancer. And so when we think about whether that has a clinical impact, we also were able to show in a separate independent cohort that higher expression of DARC, regardless of whether or not we look at all subtypes combined, or if we break them out by these different molecular subtypes of breast cancer, high levels of DARC expression in tumor are significantly associated with better survival. And so now the question is why? You know, what is DARC doing in these tumors? So preliminarily on the gene expression level, we were able to show that high levels of DARC expression in tumors also correlates with whether or not tumor-associated leukocytes were present in the tumor space. So this again is the microenvironment. So we're seeing this dose-dependent sort of correlation of when DARC is more highly expressed, we also see a higher range of tumor-associated leukocytes. Now, I'm going to get a little more into detail about that in a moment, but we also noted in that first publication that it wasn't all immune cells, but just a subset of them. And that makes sense. And so now the question is, from what we've discovered so far was on the RNA level, now on a tumor level, where are these immune cells? Are they inside of the tumor? Are they in the stroma? Are they just in the blood vessels in the tumor? And so now we use something called imaging mass cytometry. It's a highly multiplexed methodology. It's also very expensive, okay? But I'm a member of the Englander Institute for Precision Medicine, and so I get to play with all the fancy toys from time to time. And I just want to just briefly give you an overview of the technology here. What this does is utilize antibodies, which is what we would do for any diagnostic of, for instance, hormone receptor status. But now we're able to multiplex up to, this says 50 plus, but I think our boundary was, I think, around 42. And so these antibodies represent or recognize specific gene products, right? So each one of these antibodies is recognizing a different marker. They're conjugated with these heavy metal ions that normally don't occur in humans. And so by conjugating them to these ions, we can then use mass cytometry lasers to to identify the signature on a pixel level. And so the laser basically burns off the tissue, the machine actually reads the ionic signature by each pixel, and then deconvolutes that information into a pseudo image. I'm going to show you some of those. And then we're able to quantify this. So this is a molecular quantitation on a cell by cell basis, pixel by pixel basis of gene signatures, upwards of 40 on one single slide. And so that sort of removes the indirect effect of having multiple slides. We can, within one cell, identify up to 40 different markers on the protein level, which helps us with morphology, but then we also retain the spatial distinction. So where are these cells in the tumor? And so we utilize this technology to look at initially just where are the immune cells in a tumor environment that is expressing DARC or not expressing DARC. And so some of our data indicated that the immune cells that we were able to track for this first set of experiments were isolated to the stroma in a DARC low environment. So these green cells in this particular image represent the tumor, and then the red and purple represent immune cells. And so you can see that the immune cells are kind of isolated to this sort of fluid, dark space. That's the stroma of the tumor. And we don't see a lot of infiltration now. The tumor cells are sort of the cyan, green, blue color. But we see again that even the T cells and macrophages both are kind of isolated outside of the tumor environment. However, when we looked at a DARC high tumor, we actually saw that we have a lot more infiltration of those same immune cells into the tumor space. So now these blue cells represent the solid tumor, and the macrophages are in yellow. And what we also see, and this is where spatial retention of information is very important, we see that the macrophages are actually also associated with these red cells. And these red cells in this particular case are our endothelial cells. So these are blood vessels. So one of the first things you should know, and this is just a biopsy, is that the vasculature of tumors that are DARC positive, there's a lot more blood vessels in this tumor space. And where we have these blood vessels, we also see more infiltration of the immune cells into the solid tumor space as opposed to the stroma. Now, we do still see it in the stroma, but more importantly, we see it in the actual solid tumor space. And so then based on, and this is important, right? Because this also determines not only that a tumor is nearby, that an immune cell is nearby the tumor, which is what the tumor associated leukocyte score represents, but that in a DARC positive environment, it's more likely that those immune cells are not only in the stroma, but in the solid tumor space. And so that defines what the immune response is. And so the immune response is trying to get rid of the tumor, and that's true in everyone. So you know, when a cancer cell develops, your immune system tries its best to get rid of it. But using this technology, we are able to not only identify spatially where these cells are, but on a single cell basis, we're able to look at sort of what is the distribution of all these markers and how these specific cells relate to one another. And so this is a Tisney plot, and in this case, I'm just showing the ecadhedrin, which is the tumor marker. And so we see, for instance, in any given region of interest, which is represented by this white square here, we can identify the component of tumor cells versus immune cells, and then statistically use all of those markers to identify new phenotypes. And so we're able to look at the co-expression of some of these markers and identify a cluster or a signature, if you will, and then use that signature as now another diagnostic to identify if certain phenotypes of tumor cells are more prevalent in a DARC positive versus a DARC negative tumor, but then even transcending back to sort of the difference in ancestral groups, do we see a distinct tumor phenotype, tumor cell phenotype in people of African descent compared to people of European descent? And so just to recap what I've talked to you about, so we looked at DARC expression, we've seen sort of the spectrum from low to high. We see that if a tumor is high in DARC, we see that inflammatory chemokines are also high in those tumors. counteracting cytokines like CL8 are actually lower. So this will be a protective chemokine, or actually lower in dark high tumors. This is showing sort of the spectrum of immune cell infiltration across African Americans versus Europeans, and noting that there are different subclasses, if you will, or different prevalence of different subtypes in ancestry groups. And then we're using mass cytometry to further define the spatial orientation of these immune cells and what types of tumor cells are present. And so now we think about all of this in the context of ancestry. And so what's really interesting is that a few years ago, a group actually studied immune cell activity from Africans versus Europeans. And what they found was that the immune cells from Africans actually respond differently. The gene signatures were different, even if challenged with the same pathogen. And so what they is that African-derived immune cells evolved to have a unique immune response. And so this is what, this paper was actually in the context of infectious disease and in the context of just in vitro, right? And so now in the context of cancer, how much more is that going to impact a tumor microenvironment? And so I forgot I blew this up to talk about the actual signature. So they saw a stronger inflammatory response in the African immune cells compared to the European immune cells. And so this all correlates with what we've been seeing. And so this is a triple negative tumor. This actually is an African-derived tumor. And so using that TISNI designation that I talked to you about earlier, we've actually identified a new tumor type, a new tumor cell type, and we're in the process of validating this. It's not necessarily new in the sense that, you know, people have looked at this marker in blood cancers, but in the context of breast cancer, very little is known about why a tumor cell would be expressing this CD33 marker. So CD33 is actually a myeloid marker, and we're only seeing this particular type of tumor cell in our tumors that are of African descent. So what's interesting is, again, it's a myeloid marker. We normally would expect to see it on white blood cells. So if I point you to, sorry, my Zoom little thing is covering my slide here. But here, for instance, in this picture, the blue cells represent white blood cells. And so CD33 positive cells are also present in that white blood cell space, but we also see that CD33 is more prominently expressed in the tumor space. And so we see this more specifically quantified on a single cell basis, where I'm showing here the tumor cell signature. So, you know, anything above 1 represents tumor cells, and then CD33 is the myeloid marker. And we see that the tumor cells have a higher correlation and a greater expression of this myeloid marker than do the immune and stromal cell components. And so again, you know, and this is also shown here in the box plot, that the CD33 marker is actually significantly higher in the epithelial tumor cells than in the immune cells. And so again, we're very interested, right, in looking at distinctions of biology, right? So distinctions of why would a tumor cell, specifically in an African derived tumor, be expressing a myeloid marker? And what does that mean in the context of the tumor microenvironment? What does that mean in the context of DARC that's regulating these chemokines that normally act as a recruitment agent to mobilize immune cells? How are these tumor cells going to respond to these chemokine gradients if they are now expressing myeloid markers that would normally be responding to chemokines? Will it make the tumor more aggressive? Will it make the tumor more invasive? And so that's part of our future directions. I'm going to close out just talking about the fact that, you know, our ICS-BCS is a large operation. We collect germline, saliva samples. In some of our sites, we're collecting blood samples. I believe I saw my lab coordinator from Ghana log in. So this is a new process where collecting blood samples in Africa is not trivial. So I have a great team, and we wouldn't be able to do any of this work without them. But we're going to be able to look at not only tumor markers now, but circulating inflammatory markers and the composition of immune cells systemically that feed into the tumor. And now we have multiple hospital sites in Detroit through Henry Ford Health Systems, in New York through Weill Cornell. So, you know, again, this speaks to our strategic recruitment of African diaspora. New York is rich with every ancestry, if you will. And we are expanding our international sites to now include Nigeria, Kenya, Uganda, Antigua, and Mexico. And so this, again, sort of gives us the expanse, if you will, of the modern African diaspora. We partner with Dr. Olivier Elemento and the Precision Medicine Institute. He has been instrumental in allowing us to utilize not only the mass cytometry work that I showed you, but we also have access to clinical genomic testing. I didn't show you any of that data, but we also are looking for targetable mutations using the Oncomine test in our African tumors, and we have some interesting results there. We have organoids, which are basically patient-derived tumor cell lines grown in 3 dimensions. So we're able to see the tumor invasion, if you will, and this is run by our organoid team. They need to update this because I recently recruited a new member. She's an African-American, new graduate, if you will, from Jackson State. So I need to ask them for a new picture there. I need to represent my HBCU. And this is sort of a highlight of one of the organoids. I just wanted to show, you know, that even in 3D, you know, we're starting to, if you will, of tumors. And so this is going to be instrumental in looking at the microenvironment once we implement co-culture of the immune cells from the same tumors that we isolate the epithelial cells from. Part of our strategic recruitment in Ghana also includes screening for infectious markers. So this, you know, of course, when you're in the continent of Africa, you have to think about things in the context of infectious disease. And so we're going to be able to answer questions about, for instance, how some of these other infectious viral pathogens impact tumor biology. So thanks, Richard, who is online here. And then lastly, feeding all the way back to that cultural environment and comorbidity, you know, how does DARC sort of interplay in obesity and metabolism? So this is a developing project where we're looking at Duffy status. So some preclinical data actually shows that Duffy null individuals have a predisposition for sensitivity. And so all of that systemically creates a pro-inflammatory environment. And so we're going to be studying then how the pro-inflammatory environment impacts sort of the tumor environment and the architecture of the tumor microenvironment and how that changes based on not only being of African descent, but also having comorbid obesity. So in summary, I hope that I've shown you that disparities in cancer mortality can be driven by biological distinctions. We need to have genetically diverse populations. None of what I've shown you today would be possible without looking at multiple populations, without looking at admixed populations, that the immune response from an evolutionary perspective, as we look all the way back to our shared ancestry, can be very informative about how and why we see different tumor molecular characteristics of the tumor microenvironment. Looking at DARC expression, so the DARC tumor phenotype, if you will, having infiltration of immune cells as opposed to just associated immune cells, and then using these precision medicine tools, so the imaging mass cytometry, the ex vivo models with the organoids, all of this is going to be pivotal and transformative towards identifying the diversity in the tumor biology and exploiting that for better clinical outcomes. And I close all of my disparities talks coming back to the faces that we represent. And so when we look at the built environment, you know, we cannot ignore the fact that Janet from the south side of Chicago lives in a very different neighborhood than Joan on the north side, and they look very different. Janet was a young mother, and she died within 3 years of her diagnosis. Joan is a 10-year survivor and a grandmother of 5. And we have to think about all of this information that we're generating in the context of not only genetic ancestry, which is what my lens is focused on, but also how socioeconomic factors, comorbidity, health equity, all of these things feed into the biology of tumors and therefore our ability to treat and cure individuals of cancer. And these are all the people who helped me do everything I've done. And I actually need to update this slide. And I want to thank also our funding sources and the institutions that we work with. So with that, I just want to highlight some of the things from our recent Ghana trip, and I'm happy to take any questions at this time if we have time.
We have a 1 minute for questions, but first I'd just like to thank Melissa, Dr. Davis, for such a great talk. It's exciting to see all of the work that you're doing here in the United States and how that's bringing in our ancestral background to really understand where we are today in terms of breast cancer disparities. I'd like to also thank everyone for attending. We have 1 minute for questions. So
There's a pressing question.
is there
Sorry.
a pressing question that you want to ask at this point?
Who is going to ask?
Yeah, if you have a question, you can go ahead and ask. There's an incredible amount of feedback, so maybe if you want to chat your question.
I will send an email to us.
Okay.
Very nice. And I will send an email to us.
Thank you. Are there other questions? Okay, well, just thank Dr. Davis again for a great talk. Thank all of you for participating. If you're interested in receiving a recording, Melissa, is it okay for us to share that with folks?
Sure.
Okay.
Yes.
And if you do want to get access to the recording, please contact Erica Jenkins to make arrangements. And thanks to Erica for making all of this possible. So one question. So I think everyone sort of, the consensus says, fantastic talk. Thank you so much. So we'll let you go. Thank you, everyone.
Thank you, everyone.
Bye-bye.
ভালো বলেছে কিন্তু ওরা যে ধরনের কাজগুলো করে সেটা হচ্ছে ওই রকমই কাজ আর কি যেটা couple of genetics কে নিয়ে জেনেটিশিয়ানকে নিয়ে এসব নিয়ে চলো খাবার